Hey guys, so in this video we're going to be looking at antigen presenting or antigen presentation. It's the step between the second and third levels of the immune defense system. So our learning intentions for this video are to know the purpose of antigen presenting, to understand the role of the MHC markers and compare the difference between MHC1 and MHC2. So what is it? So antigen presenting is the way that our second level of defense is able to communicate to the cells of the adaptive system, so our T and B cells. And basically it's how our second system goes through to the third level of the immune system. So the way it works is our cells are covered with these surface proteins known as major histocompatibility complex proteins, quite a very long name, but we can shorten it to MHC. And they're used to display antigens on the cell surface. They're sort of like, I guess, a little marker for the immune system cells to look at. And there's two main types, MHC1 and MHC2. And that 1 and 2 is to do with its structural makeup as well. So here you can see MHC1 and MHC2 markers. So our MHC1s are pretty much found in every single cell in our body, and they're used for a variety of reasons. We show our self antigens on them. So again, if we're displaying to the immune system that we're ourself, it will show the antigens on MHC1. If a cell is infected, it will actually start to show some of the pathogen on MHC1. And if a cell is cancerous, it's actually going to show antigens to indicate that it's cancerous, and again, it's showing them on MHC1. So the three main reasons we use MHC1 is for self, infection, or cancer. And these are checked by an immune system cell called T cytotoxic cells. So they come along and they scan the MHC1 markers and determine, okay, is this a self cell? If it is, we can leave it alone. If there's an infection or if there's cancer, the T cytotoxic cell will actually initiate apoptosis, which we've learned about a few lessons back. So basically, MHC1 are checked by the T cytotoxic. And I've got a bit of an example here. So imagine this is just an everyday cell that's become infected. You can see it showing off self antigens on MHC1 at the moment, but when it becomes infected, it's also going to start showing off these viral antigens, okay? And now the T cytotoxic cell is going to start scanning, and it sees the MHC1 with self, and that's fine. But when it detects the viral antigen on MHC1, it's going to start to release some cytokines, which we know are just signaling molecules, and it's going to start to cause apoptosis in that cell, and we get rid of that cell. And that's how we can actually stop cells that are infected or cancerous from being able to spread, is by using MHC1 and using T cytotoxic cells. MHC2 is, I guess, more specialized marker, and these are only found on cells known as antigen-presenting cells. And so the three antigen-presenting cells are dendritic cells, macrophages, and B cells. So these guys also have MHC1, but they're the only cells that have MHC2. And the reason why they have MHC2 is because these are our ones that will go around and engulf a pathogen or an antigen and will display it on the outside of its cell. Even if it's not infected, it's going to start showing antigens from the pathogen that it's engulfed. So you can see in this diagram, here we have what looks like a macrophage, and it's engulfed a microbe or a bacteria, and it's going to ingest some of those antigens and start to display it on the MHC2 markers. So it's a little bit different, it's MHC2, and those MHC2 markers go and activate T helper cells. And we'll go on to what T helper cells do a little bit later on, but basically they're involved in helping the rest of the immune system work. So they're really, really important that we have to activate them first. So again, this could have been a B cell, this could have been a dendritic cell, but any cell that engulfs and then shows off will show it off on the class 2 MHC molecules. So again, a quick little example. Here we've got a pathogen, and this is an antigen presenting cell. So it could be a macrophage, could be a dendritic cell, could be a B cell. It's going to engulf the pathogen and start to break it down with some lysosomes into smaller fragments known as antigenic fragments. And it's going to then display those fragments on the MHC2. You can see that this cell still has MHC1. Now that would be used to maybe show off its self antigens, but the MHC2 fragment now is showing, hey, there's something around that isn't meant to be here. And that's going to go and activate a T helper cell. And the T helper cell then is going to go and initiate a wide range of things that sort of activate other relevant cells in the immune system. So again, MHC2 is for when they've been engulfed to go and activate T helper cells. So here's, I guess, a video of what it might look like. So there's our pathogen being engulfed. It's broken down and displayed on the MHC2 markers on the outside. And that would go and activate those T helper cells we talked about. So in review, nearly all of our cells in our body have MHC1, and they're used for self-antigens, infections, or cancer. And the MHC1 are checked by T cytotoxic cells. Now the APCs are the additional MHC2 proteins. So they're the only ones with MHC2, and they present engulfed pathogens or antigens to T helper cells using those MHC2 proteins. So a lot to get in this video, but the basics is to know the difference between what MHC1 is used for and what MHC2 is used for. So I hope this video helped. Thanks, guys.